Students, in this class we are going to study some of the important diseases affecting sugarcane. And these are all the list of diseases which have been already recorded on sugarcane causing economic yield loss. The first and foremost disease is the red rot caused by Coltotrichum falcatum, and then the smut caused by Ustilago cytomania, which is recently renamed as Sporisorium cytominium, and then set rot or pineapple disease caused by Ceratocystis paradoxa, wilt caused by Fusarium saccari, rust caused by Puccinia erianthi, and Pocoboyong. It is a newly emerging disease caused by Fusarium moniliforme, and then some of the bacterial diseases which includes gummosis, red stripe, as well as ratoon stunting, and besides the phytoplasma causing grassy shoot disease and the virus which is causing yellow leaf disease, which are all important on the sugarcane. Let us see one by one in detail along with the management aspects. And first and foremost is the red rot, which is caused by Coltotrichum falcatum. In India, it was first observed by Barber in 1901 itself in the cultivar Red Mauritius in the region of Godavari Delta of Andhra Pradesh. Later, Butler in 1906, who conducted extensive studies and worked on the causal organism, its epidemiology, etc., and he coined the term red rot. And what are all the predominant symptoms? The external symptom that appear on the third or fourth leaf, and the crown may wither off, and reddening of internal internodal tissues with the crosswise white mycelial growth, and then the stalk become hollow and the rind shrinks longitudinally with the black velvety fruiting bodies, and then it also produces minute red spots on the midrib of leaves, and this is the yellowing and drying of the third and fourth leaves of sugarcane, and finally leads to drying of whole clumps. And when the disease is severe, the entire field become completely dried and leading to great loss, yield loss. And the externally upon the canes, the internodal discolorations and the nodal sporulation as pinkish powdery spores formation are the predominant symptom on the canes. When you take the longitudinal section of affected canes, you could see the crosswise white mycelial growth which are interspersed on the affected canes, and then the cavity become a cavity will be formed, which is irregular cavities formed on the affected canes, and these cavities are filled with fungal growth. And the pathogen etiology and epidemiology of the pathogen was studied by E.J. Butler, and the sexual stage of the red rot pathogen which was identified by Kona and Srivastava, and it produces acerulae on the all infected plant parts, and this acerulae which contains conidia, conidiophore and setae. Here the conidia, which is a falcate shaped hyaline single celled, and the setae are long and rigid in nature, septate, and it appears like a bristle, and it appears like a bristle, long septate and appears like a bristle, and the fungi is set bone, and it survives long in the infected clumps. The rain splash or the air current which helps in spreading of the disease. Monoculturing of sugarcane and successive ratooning of sugarcane, which is responsible for more incidence of which is responsible for more incidence. Monoculturing and successive ratooning causes more incidence of red rot in the field, and there should be a comprehensive effort taken for the management of this disease, which include cultural practices, physical as well as the chemical methods of applications. In the cultural, you have to select the healthy sets, disease free, from collected from the disease free area. And if the incidence is very high in a particular field, then go for crop rotation with the non-host crop like rice, and then use resistant or moderately resistant varieties like say co 86032 or co 86249 or co si 95071, cog 93076, etc., have to be used in order to reduce the incidence level. And then physical method that includes complete physical removal of affected clumps at an early stage to avoid further spread of the disease, and then treating the seed set in the aerated steam therapy, and subject to aerated steam therapy, and keep the sets in the machine at 50 degrees Celsius for 1 hour, or using the hot water treatment, that is treating the heart with the hot water at 50 degrees Celsius for 30 minutes or 52 degrees Celsius for 18 minutes. While doing these operations, hot water treatment or aerated steam therapy, the temperature and the time specified should be strictly followed, which will be helpful in maintaining the germination potential of these seed sets. And then set treatment with the carbendazim 0.1%, that is 1 gram ml mixed in 1 liter of water, and along with the 2.5 kg of urea mixed in 250 liter of water, which will be helpful in reducing the inoculum density as well as enhancing the germination. And use of some of the fungi toxic chemicals like benomyl topsin, which is not, and these fungicides are not available. And the chemical method, that is set treatment with the carbendazim or spot drenching during the growth period, that is also advisable. And this carbendazim is responsible for reducing the inoculum density in the seed set material, and this urea helpful in hastening the germination and growth process. And then second important disease is whip smut caused by Sporosodium cytaminium. Earlier it was known as Ustilago cytamina, and it is renamed as Sporosodium cytaminium. This disease was first reported from South Africa, and this is otherwise called as whip smut as well as culmiculus smut, because the entire culm region is converted into long smut sore, and hence it is mentioned as culmiculus smut. Production of a whip-like structure from the growing point of the canes, which is covered with a translucent silvery membrane, which encloses black powdery mass of smut spores. Under the in the affected plants, the internodal length is got reduced, and profused sprouting of lateral buds with narrow erect leaves are also noticed in the affected plant. And the smut spores, which are spherical in shape, light brown in color, and minutely accumulated on the outer wall. This is the typical smut incidence. Since the entire central shoot is converted into a long smut sore, it is mentioned as culmiculus smut. And these are all some of the allied symptoms. As I mentioned in the previous slide, the leaf size got reduced and they were rigid, and proliferation of axillary buds are noticed, and then this is the malformation of flowers, and then the floral infection and proliferation of lateral buds and bud infection. And here also we need to practice comprehensive measures to control this disease. Among the cultural methods, that's growing of resistant or moderately resistant varieties, which includes CO 86249 or COG 93076, etc., have to be used, and discourage ratooning of diseased crops which are having more than 10% of infection, so that the infection ratio or the disease spread got much reduced under field condition. and then growing the Cajanus Cajan red gram as a companion crop between two rows of sugarcane, which reduces the secondary spread of this disease. And among the physical methods, the set treatment, aerated steam therapy. The subject the sets to the aerated steam therapy at 50 degrees Celsius for 1 hour, or the hot water treatment. Keep the hot water at 50 degrees Celsius and immerse the sets for 30 minutes, or 52 degrees Celsius for 18 minutes. So as I mentioned earlier, here also you should maintain in all the cases. The vegetatively propagated sets have to be, if you subject to the aerated steam therapy or hot water, the time and the temperature has to be maintained exactly, otherwise it will affect the germination potential of the sets. And the rowing out of the smut or affected whips in a gunny bag and then dip it in the hot water for 1 hour to make the spores inactive, or uproot the affected plant, completely affected plant, to reduce the further spread of disease. And among the chemical methods, treat the sets with some of the fungicides like triadamifone or carbendazim mixed at the rate of 1 gram per liter, and soak the sets for 10 minutes and then use it for sowing purpose. And then the next important disease is set rot of sugarcane or pineapple disease of sugarcane caused by Ceratocystis paradoxa. Why it is called pineapple disease? The affected canes that emit a alcoholic odor which may resemble the odor from pineapples, and hence it is named as pineapple disease. This disease is observed as soon as the sets are planted. So the infection, which leads to poor germination of sets and showing some gappiness in the field, yellowing and drying of emerging leaves are the common symptoms. And it also produces reddish brown discoloration inside the affected sets with pineapple odor, as I mentioned earlier. Blackening of sets with profuse fungal growth are seen in the advanced stage of infection. This pathogen, Ceratocystis paradoxa, it is known to produce two types of spores, macro as well as microconidia. These macroconidia are exogenous, that is produced outside, which are single-celled, olive green or black in color, spherical or elliptical in shape, while the microconidia, which are thin-walled and cylindrical, black in color and produced endogenously within the conidiophores. And these are all the typical symptoms. What are all the conditions that, as I told you, the internal discoloration which turn into pinkish or reddish discoloration, emits the pineapple odor, and it forms the cavity inside which is completely filled with mycelial growth. These are all irregular cavities. This is the healthy cane, longitudinal section of the last one is the longitudinal section of healthy cane, and these are all the sporulating structures produced by the pathogen. This pathogen produces typically a long necked ascomata, and this is the long necked ascomata, and which produces the osteolar hyphae, through which the conidia are released, and this is the barrel shaped conidia, and these are all the hat shaped ascospores. And this, as we know, we might have studied in while we studying fusarium. This is the typical terminal chlamydospores produced by the organism, and these are all the conidiophore as well as the phyllates which bears the conidia. And the ill-drained field or heavy clay soils where where the drainage is the problem, the disease will also be very severe. And the wind-borne conidia as well as spreading of conidia through irrigation as well as rainwater helps in spreading of this disease. And use healthy seeds collected from the disease-free area and plant it in the field which are having proper drainage and completely sanitized. What do you mean by field sanitation? Removal and destruction of the residues of previous crop. Mostly the sugarcane is grown year after the year, and most probably the previous crop would also be of sugarcane, and those infected plant debris have to be removed from the field to avoid the initiation of the disease. Before planting, dip the sets at 40 ppm of boron or manganese and as well as spraying of the same components which will reduces the disease intensity, and treat the sets with the carbendazim 0.1% solution for 10 minutes. That will helpful in reducing the set bone inoculum. And when during the crop growth, if the incidents are noticed, as I told you, which affects immediately after the planting. So after the planting, if it is noticed, then you can go for drenching with the carbendazim. And then the rust disease, which is common in many of the crops. Here also the Puccinia erianthe, which causes small elongated yellowish spots initially, which appears on both the surface of the leaves. Later the spots increase in length and turn into brown or orange brown or reddish brown in color. These sori or iridosori are coalesced together and form larger irregular necrotic area. It shows the rusty appearance of this disease, and this eventually or ultimately leads to premature death, drying and death of leaves. The pathogen known to produce thick-walled orange-brown accumulated iridospores with equatorial pores for germination. And then the teleospores, which are bicelled, dark brown in color, clavate in shape, slightly constricted at the septa, and it survives in the infected plant debris as well as stubbles and the other alternate crop like Saccharum spontaneum. And for managing this disease, grow the resistant varieties such as Co 91010, that is Dhanush, or Co 87025, that is Kalyani, are recommended. Remove and destroy all those infected stubbles from the or plant debris of previous crops. And then application of mangoseb or triazole or strobilurin are recommended to manage the disease at the field level. And then the wilt disease caused by Fusarium saccharine, which was first appears in the crop which are all 4 to 5 months old. Gradual yellowing and drying of foliage, shrinking or withering of canes are the noticeable symptoms. And then the pith, internal pith, turn into light to dark purplish or brown discoloration. And pithiness or boat shaped cavities or spindle shaped cavities formation, and in between the internodal region are the characteristic symptom. And this infection also emit a disagreeable odor due to severe infection by this pathogen. And often you may also see the white mycelial growth on the pithy region. This is the irregular cavities with the purplish border or discoloration, spindle shaped cavities filled with mycelial growth. And in severe case, pithiness are the common symptoms of affected canes. And as we know, this Fusarium is known to produce macroconidia, microconidia and chlamydospores. Here the microconidia are single-celled, hyaline and oval, but not in this case the macroconidia are long, falcate shaped or sickle shaped and with 3 to 5 septation. And use the healthy sets as mentioned for many other diseases. And the cultivar Co 617 is found to be resistant to this wilt disease. Crop rotation, managing the root borer and avoiding the prolonged drought as well as the waterlogging condition and hygienic practices are also recommended to reduce the incidence under the field level. As mentioned in the previous disease, here also dipping the sets at 40 ppm of boron or manganese or spraying with any of these elements which reduces the disease intensity. Set treatment with the carbendazim at 0.1% and spot drenching with the carbendazim at 0.2% are also recommended to reduce the incidence at field level. The next one is the Pocoboyang disease. And this disease is a typical disease producing malformation or abnormalities on the top portion of sugarcane plant, and this is caused by Fusarium moniliforme. And this pathogen is known to produce 3 important stages of infection or symptoms. What are all they? That is a chlorotic phase, and then acute phase, and during this acute phase the top rotting is the common phenomena, and then third one is the knife cut phase, and this is associated with the second phase itself. When you remove the affected sheath, you could see the knife cut symptoms. And then first one is the chlorotic phase 1 and 2. This earlier symptom of this disease is the chlorotic phase, and the chlorotic condition towards the base of young leaves and occasionally on other parts of the leaf blades are noticed. Frequently pronounced wrinkling or twisting or malformation and shortening of leaves accompanied with the malformation or distortion of the young leaves are noticed at the base of the spindles, affected leaves which become narrower than the normal leaves, and in the matured leaves it produces irregular reddish stripes or specks within the chlorotic area, and later these necrotic or reddish turn into reddish in color, and similar type of specks or streaks could also be seen on leaf blades and as well as on the leaf sheath and midribs. And this is the acute stage, or otherwise called as top rot phase. The most advanced and serious stage of infection is the acute phase or the top rot phase. The young spindles are killed and the top portions completely dried and dies. The plant dies. In the advanced stage of infection, malformation of the spindle as well as the pronounced wrinkling, twisting and rotting of spindle leaves are common phenomena, and the formation of red specks or stripes on the whole base of the spindle, and affected spindle gets rotten, dries up very fast, and finally it is termed as top rot, and the tender tissues of the apical part become completely rotten. Sprouting of buds are also observed in the severe case of infection, and in such case the apical parts of the stalk is seriously damaged. And then the third part, which is associated with the second, that is acute or top rot phase, and in this case you can see the knife cut symptom. The symptoms are seen on the affected portion. This symptom or this knife cut stage is observed in association with the previous stage, that is acute stage or the top rot phase, and it is characterized by one or two or even more transverse formation of transverse cuts on the rind of stalk or the stalk or of the stem or the stem. In such case, uniform manner and the tissues are removed, which will be resembling that the tissues are removed with the sharpened knife, and this is the most damaging stage, and the typical ladder like lesions. In the previous slide I shown you, this is the typical ladder like deep cuts are formed due to severe infection. When you remove the leaf sheath, you can see the horizontal conspicuous cuts are seen on the stalks, and these are all the different symptoms produced by the pathogen chlorotic phase 1 and 2. In this case, formation of the reddish brown streaks on the leaf and leading to rotting and drying of leaves. This is the rotting of top portion, and this is the ladder like knife cut symptoms beneath the sheath of affected canes. And the incidence will be severe when the temperature ranges from 20 to 22 degrees Celsius prevailing, and therefore the disease is more severe, and hence this type of temperature could be achieved only during the low temperature period, might be influenced by the rainfall, and then the pH of 6.5 to 7.5 in the soil, which is also helpful in spreading of this disease, and average humidity of 70 to 80% or even higher with the cloudy weather and drizzling rain are favorable for the development of this disease. It is primarily transmitted through the air currents by the conidia, and then secondary transmission is through the infected sets or through the irrigation water or rain splash. The best means of controlling this Pocoboyong disease is growing resistant varieties. Avoid susceptible varieties such as CO419, MS 10001 and COC671 likewise. You should avoid the varieties which are reported as susceptible to this disease. And then spraying of 0.1% carbendazim or 0.2% copper oxychloride or 0.3% mangozeb. Minimum of 3 sprays at 15 days interval are recommended to reduce or to control further spread of the disease. And then canes showing the typical top rot or knife cut symptoms should be rogued out to avoid further spread. And there are a few bacterial diseases are also important in the sugarcane. First one is the red stripe disease caused by Pseudomonas rubrilinians, which produces water-soaked elongated streaks which later become chlorotic and reddish brown in nature, and two or more stripes coalesce together and form larger bands and killing of growing points as well as terminal portions and leaves. The bacteria, Pseudomonas rubrilinians, which is rod-shaped and motile with 1 to 3 polar flagella. The primary spread of this disease is mainly through infected canes, and secondary spread is through the rain splash and irrigation water. And some of the cultivars are reported to be resistant to this red stripe disease, which are listed here. That is CO 6805, 7202, 7321, etc. And avoid the collateral host near the sugarcane field in order to avoid severe spread of this disease. And some of the insects may also act as a vector in spreading the bacterial bodies from infected to healthy in order to reduce the insect mode of transmission or spreading. You have to apply effective insecticide to reduce the insect population. And then the gummosis symptom in the sugarcane, which is produced by the typical bacteria Xanthomonas axinopodis, Pathova vascularum, and upon the matured leaves it produces longitudinal stripes or streaks around the veins, especially near the tip of the plant. Initially these stripes are pale yellow, later turn into brown and dry up, as we studied in the previous Pseudomonas rubrilinian symptom. And the affected canes become stunted, shortening of internodes, giving the bushy appearance. The longitudinal splitting of sugarcane is common, and which shows the exudation of dull yellow bacterial oozes. The fibrovascular bundles are deep red in color, and the internodal cavities are also filled with yellow colored bacterial oozes or gums. The pathogen, that is Xanthomonas axinopodis, which is a short rod-shaped gram-negative non-spore-forming bacteria with a single polar flagella. The first and foremost thing, the affected plants or plant parts should be completely removed and destroyed, and the field should be free from previous infected stubbles. And then select disease-free sets from the disease-free area, and avoid the collateral host like maize, sorghum, or pearl millet, etc. near the sugarcane field. And among the viral diseases, the tobacco mosaic disease is common, which is predominant and prominent on the young leaves, which is caused by tobacco mosaic virus, which produces linear chlorotic stripes alternating with dark green areas on the leaves. Later, yellow stripes appear on leaf sheath as well as on stalk also. The virus which is producing the disease are rod-shaped, which belongs to potato virus Y group, and got transmitted through the infected sets as well as through the aphid vectors Rafalocyphemidis, Melanophis saccari, Melanophis indosaccari, etc. It also affects maize as well as sorghum plants. So select healthy sets for planting, and then subject the sets or propagative material to aerated steam therapy at 56 degrees Celsius and for 33 hours, and then hot water treatment at 52 degrees Celsius for 20 minutes, helpful in reducing the inoculum in the vegetatively propagated materials. And then the grassy shoot of sugarcane, which is a phytoplasmal disease, and the infection which causes production of numerous lean and lanky tillers, and the leaves become completely chlorotic, thin and narrow and crowded at the top, giving a bushy appearance. And this crowding of leaf is especially due to shortening of intergrowths. The reduction in the leaf area, which gives appearance of grass like The leaves of affected plants are looking like gross leaves due to infection by this phytoplasma, and it got transmitted by the aphids Melanophis sakari. There are some varieties said to be resistant to this grassy shoot disease, which includes CO86249 or COG93076 and COC22. Avoid retuning of cane, which helps in reducing the disease incidence. When the disease incidence is more than 15%, avoid retuning, so that the further development of disease and spreading of disease could be avoided. And then the, the symptoms that appear within 2 weeks after the planting. If you notice at the early stage and replace the affected seedlings with the healthy seedlings, these uprooted should be disposed of properly. Rogue out the affected plants, affected plant seedlings, and then treat the sets are subject to the aerated steam therapy at 50 degrees Celsius for 1 hour, or treat with the hot water treatment for 54 degrees Celsius and prolong it to and do it for 8 hours duration. And then spray some of the pesticides effective against the sucking pest. The pesticides like dimethoate or methyl dermaton can be used to reduce the vector population. Avoid retuning if the disease is noticed and the incidence is more than 50%. And then ratoon stunting, it is also another unique disease which is caused by the xylem inhabiting fastidious bacteria Lifesonia xyli, subspecies xyli. So the infection that leads to stunted growth of the affected plant, reduced tillering, and then thin formation of thin stalks and shortened internodes and yellow foliage. It also produces pinhead like orange discolored spots or dots of bacteria on the internodal tissues due to bacterial building up of bacterial population. And the pathogen mainly present in the xylem area and transported to the plant system. Within the plant system through xylem vessels, use healthy sets collected from the healthy area, and then field proper sanitation is also recommended to avoid infection from the debris of previous crops, and then remove and replace the ungerminated sets, and treat the seeds with hot water at 50 degrees Celsius for 2 hours, which gives 100% control over the infection. And aerated steam therapy is also hold good to reduce the set bone inoculum. And then use the chemical disinfectants to disinfect all those implements, cutting knives, etc. And at least 5 minutes of contact with the cutting surface is needed to assure disinfection. You should not just simply wipe, and it should be completely wiped and keep it wet with the disinfectants for a set period to keep these implements away from the infective propagules. And then yellow leaf disease, which affects the crop at 5 to 6 months. Yellowing of midrib patches as well as the adjacent laminar region is the predominant symptom. Subsequently, the affected leaves become dried and the midrib 3 to 5 leaves from the top. Then later it spreads to all the leaves. In some cases, the red discoloration may also be seen. In severe cases, drying of spindle, the topmost portion, is also possible, and this infection leads to stunted growth of the cane, and then reduction in the internodal area. And this disease can be identified from a long distance due to bunchy appearance at the top of the cane and shortening of shortened and stunted growth of the plant. And these are all the various symptoms just expressed explained in the previous slides. And the previous slide, this is the yellowing of leaf, yellowing of midrib, and drying of entire plant or all the leaves, and this is transmitted by the aphids Melanophis sakari and Rafflosipum maides. And this virus SCYLV, which is coming under the Litioviridae family, which causes localized infection especially in the phloem cells of the plant. And for the management of this disease, you collect the disease free sets from the disease free area, and maintain proper hygiene of the field, use resistant varieties, and avoid secondary spread through the insects by spraying the effective pesticides against the sucking pests. And one more severe problem is the growth of a phanerogamic parasites, that is Striga euphrasioides, in the field, which also a menace in recent days, which is a partial root parasite and growing by attaching itself with the roots of a sugarcane plant, and it form leafy shoots just at the base of infected canes. The way parasite affected canes become yellow in color, and it since the striga which absorbs the nutrients and water from the host plant, and causes yellowing and drying and lean lanky canes due to complete absorption of nutrient and water by this phanerogamic parasite striga. And this can be controlled by application of 2,4-D at the rate of 2.5 kg per hectare mixed in 500 liters of water. And then iron chlorosis, which is a nutrient deficiency. It is also one of the common problem in the sugarcane cultivated area. The young leaves wear the pale stripes and scanty chlorophyll content that appears parallel to the lines, and these leaves completely turn white and even, including the veins and veinlets and midribs. The deficiency that leads to restricted root growth, stunted growth, and constriction of internodes, which are all the allied symptom by which you recognize the iron deficiency. So, in order to get away from this problem, spray ferrous sulfate at the rate of 250 to 500 grams mixed in 100 liters of water, or even soil application of 25 kg of ferrous sulfate per hectare, or spraying of 5 kg of ferrous sulfate along with 2.5 kg of urea mixed in 500 liters of water for 1 hectare is also suggested to get away from iron chlorosis problem. The foliar spraying has to be repeated with the interval of 7 to 10 days